did all the computer courses last night, and I was like, she's.
Yeah, the early,
Unfortunately.
yeah. Yeah. I wish I could definitely just make to like, but then the teacher in me is like mean.
Yeah.
So
8 weeks is just rough.
Are these ones
I wish
pretty far, like the ones on the?
I don't think they are. I think that the midterm is more difficult than these, but that's why I'm saying if you guys don't do well on these, and you know that you really need to go back and study, especially whatever chapters. This is more like trying to figure out, you know, what you're doing terrible with. Okay, so I've got chat pulled up because people have a tendency that when they're on the Zoom to chat instead of talk over it, which is fine. I'm not very good about looking at it. So if it gets in the way, minimize it. Okay, so I guess it's only gonna be 5. And remember, so I'm always trying to be over here because the microphone's over here. It's hard to like be between. But okay, here we go. Here's more. People can always join in if you haven't joined in yet. People can always join in as we go as well. This should have the game pin up on the screen. So I'm going to go ahead and start it. I'm not going to lock it. Unless I'm not doing that. Yes, yeah. After we do this, when I get around to posting it after this, it won't be long. All right, so for our lecture midterm, we're going to have 4 for the review. Anyways, we're gonna have 4 questions per chapter. And then after that, we'll have bullet points of more to study that didn't get covered by those 4 questions, obviously. Even then, everything that I'm saying there is not technically comprehensive. I just don't want anybody leaning on that and being like, you didn't cover this in the review. Yeah, I'll just be like, yeah, I know. No, but it's happened. That's why I say. So use this as a tool to tell you what you need to focus your studying on. Okay. Again, so this is me apologizing because I can't see the mouse over here. All right. This will be chapter 1. This is our intro to microbiology chapter. All right. The theory that says all living things are made from cells that originated from other cells is known as, let me move this. I'm sorry. Oh, good job. So we're off to a good start. Ready. So how would you define microbiology? Okay, so yes, you guys all got that one too. Good. Let's just think about how, but I can click next for these. So I was just trying to make sure that if we get to ones that like people really mess up on, that I have time to explain it. So obviously you guys don't need this stuff explained. So figuring out now is better. It doesn't go automatically now that I'm saying. All right, which of the following are not products that are made thanks to microbes? This is multi-select, it could be more than one. Also could be a trick, but it might not be a trick. The multi-tube. Luckily, just that won't be on the midterm. It'll just be one. So it says which of the following are not products that are made thanks to microbes. It's supposed to be that they all are made thanks to microbes. So it should have been the red one, right? Not products made.
No, it would be the purple. Yeah.
Which
all
is
of
fun.
these- All of these are, yeah.
Well, all of them, okay, yeah, because of the wording of the choice,
Yeah,
I
yeah.
guess. It's a weird, yeah. Luckily, this crap isn't gonna be on there, because it's definitely weird. And I'm sure the way you guys are thinking about it was how I wrote it at the time, but now when I'm looking at it, all of these are, and then to me, I'm looking at all of these are not products, or all of these are
I
products.
think it's R, well, mostly because at least 3 of them are obviously.
Well, yeah, so I guess I'll just clarify, because we're talking about it, that all of those are. All of them are products made by microbes, including the acid, so. Okay, next. It's just not set, that one wasn't set up right, that wasn't right there. Okay. Which one has the correct order of taxonomic classification from largest at the left to smallest at the right?
My thing is over.
Yeah, that happens with this. That's why I don't do these for credit, because some of it just doesn't work anymore for people. Yeah, you should be able to get out of it and then reload it and join back in with the number that I'm covering down at the bottom. Get this out of the way. There we go. Okay. Okay, so our answer is going to be phylum, class, and order. The most popular one that you guys picked was that kingdom, phylum, and order, because we're missing, I guess that is correct, kingdom, phylum, class. It is technically correct. They're both right, but nobody picked the blue one either. So.
Kingdom like that.
Yeah. That one would have, I guess, kind of, because I was trying to find a different one to put in as a choice. Now I'm realizing that that was not as sneaky as I thought it was. But whatever, it's not for Friday. So that's it for chapter 1. That's all we're going to go over. We all know chapter 1 is going to be easy, right? So that's child's play. Chapter 1, also, you're going to want to know germ theory, which was developed by Lister. And that's a concept that says that if you have a communicable disease, it has to be caused by a germ of some kind, of microbe of some kind. There's no spontaneous generation. It doesn't come out of nothing. So that's all germ theory says. Binomial nomenclature, that term and what that actually means. So that's like Escherichia coli, and how to properly write it with italics and which one's capitalized and all of this. So that is chapter 1, easy stuff. Chapter 2 is going to be our chemistry review. I'm actually going to close this because I think it'll be easier to. Organic molecules are any compounds that contain carbon, true or false? All right, so it's going to be false. It has to have carbon and hydrogen. So you guys know. Cool. So where does the number that we assign for pH actually come from? So the answer is that it's an inverse of the hydrogen ion concentration, and it is logarithmic, so it's based in 10, but it's still an inverse of that concentration. So when we look at a pH of 2, what that means is it's 10 to the -2 concentration of hydrogen ions. So if you have a pH of 10, it's 10 to the -10 concentration of hydrogen ions. So which one's smaller, 10 to the -2 or 10 to the -10? -10 is smaller, yeah. So that's how you get those numbers and what they actually mean. I have 2 different questions that you can get about because I have, it's gonna be different for people, about pH. So I'm just like giving you guys a heads up about it, that one of them actually involves it's not hard math or anything, but just like knowing how the math works, essentially. If you don't understand how this math works for that pH, where we get 7 from, then yeah, you're not gonna get that question right. So be aware of that. Okay. I was just trying to think how I would phrase it, but we'll just leave it alone for now. So what kind of bond involves a true transfer of electrons from one atom to another? So we're talking about a transfer of electrons. So most of you guys got this one. Transfer of electrons, like from moving it from one atom to the other atom, that's going to be ionic bond. So if they were sharing it, that would be covalent. And remember, hydrogen doesn't have any exchange of electrons. So which level of protein structure is defined by the actual amino acid sequence itself? Right, exactly, that's going to be primary. So primary is the amino acid sequence. Secondary is alpha helices and beta sheets. Tertiary is going to be whenever we have interactions of the other R groups in there, causing folding over and even disulfide bonds that are covalent bonds across from one amino acid to the other. And then quaternary is when we have like the subunits coming together to have the functional protein. So just to review that real quick. Alrighty. Okay, so also for chapter 2, you're going to want to know, covalent and hydrogen bonds, which we kind of just talked about. Functional groups in organic molecules. I don't want you to go too crazy. I'm not going to ask you about esters or something, but I would be aware of what that group would be. That's a useful one to be aware of. Like it's OH, hanging out on the end by itself. So those ones, phosphate group, that's another functional group. So just be aware of those ones that we've talked about kind of repeatedly that sound familiar. If it sounds familiar, you should probably know what it is, clinically. Polarity. Biological macromolecules, that's just going to be, proteins and carbohydrates and nucleic acids and the lipids, those four. And what they are and what they do for the cells. Going to have polarity. That's going to be, as long as you understand why something would be polar, actually has to do with the electronegativity of like oxygen or something. It hogs those electrons to it. So it would be negative because it has more electrons associated around it more often. And then that leaves the little hydrogen with positive. So that's what gives the hydrogen positive, charge polarity to interact with negative atoms. And in water, in the actual solvent of water, those interactions between the positive hydrogens and the negative oxygens are kind of what gives it its structure and why you have surface tension and stuff like that with water. Okay, and we did bring up beta sheets and alpha helices. Those were in that secondary structure for proteins, but if you don't know what that means, you might want to familiarize yourself with that. So, which is structures of the protein. All right, moving on to chapter 3, we're going to talk about the tools of microbiology. What type of media allows some microbes to grow and inhibits the growth of others? Great job. I didn't want to say anything, but it's written on the board. So that's what we're doing when we get back. So yes, great job, guys. I feel like with that stuff, a lot of people have trouble with that. So when attempting to isolate a microbe from scratch, where would you start? Be the first thing that you should probably do. Good job. Yep, that's going to be a straight plate. So we can isolate it out, right? Moving on. Which part of the microscope allows you to control the amount of light being focused on the specimen slide? That brings me to another thing that I wanted to bring up, which is that obviously there's overlap between lecture and lab. I did not check to see if the questions same thing. So I wrote them completely different times. So that it's possible that you might see some of the same exact questions, but just be sure that you read over them and make sure that you are actually reading the right thing and not like doesn't have a knot in it or something like that. All right, what is the purpose of alcohol in the gram stain? Right, and this one is a difficult one for people. Everybody knows what crystal violet's for. Everybody knows what saffron is for. So iodine, remember that's going to be our mordant. It's going to cause the crystal violet to get stuck in place. And then after we get it stuck in place, then we will rinse with the alcohol. And it's not meant to rinse away the iodine. We do that with water first. What it's meant to do, the alcohol is rinsing away the crystal violet. So that's what it's actually rinsing away. So it's a decolorizer. So if you're gram negative, you will rinse out all of that crystal violet because you have a spindly little single peptidoglycan layer. And then you go back and stain with saffron and see red. Does that make sense? Okay. Cool. All right, so that's what we're covering for this part of chapter 3. Remember, we're going to need to remember what a pure culture is. That's just like it's one organism in there. So it's not like you swabbed the floor and then grew it up and then that came from one source and it's pure. That's not how that works. You want one organism. Fastidious and growth factors. If an organism is fastidious, you will want to grow it on enriched media, which has growth factors in it, because those are the things that they can't produce for themselves and it has to be provided to them in the media. All right, the different kinds of media, I mean, you have selective, you have differential, complex. That is a term referring to, that's one I feel like people don't understand very well when they see it. That complex media is referring to, it has something in there, we don't know the exact composition of it. So whenever we have like brain heart infusion auger or something like that, we don't know exactly what's in the brain heart that we're putting, but we know it's brain and heart from some organism. So something that we don't know the exact chemical composition of, that would be complex. But yeah, so those different kinds of media and what they might be used for. Microscope function, purpose of oil immersion. I'm not even going to go over that. You guys know about this stuff. Different types of stains and their purpose, same exact things, what you need to know for lab anyways. So you guys should have an idea of what you need to know for that. Okay, moving on. Chapter 4, this is dealing with the prokaryotes. Which prokaryote has thick peptidoglycan wall studded with lipoteichoic and teichoic acid? Yep, good job. I'm glad that is working out for you guys. That's one of the things I feel really bad about if we didn't get. All right. Cool. Okay, didn't miss anything. All right, next question. If you're covered all over with flagella, what flagellar arrangement would this be? All right, so the word for that one is peritrichous. I know that's more or less just a vocab thing that you guys need to study on. This didn't remind me that I wanted to, now, and apparently didn't remind me enough because I already forgot what it was. Sorry. I will remember what it is. Don't worry about it. I had to do just the general test, so I'll remember it. All right, so cool. What is endotoxin? That did remind me, actually, so I'm glad that. All right, so endotoxin is part of the gram-negative envelope. So it's the outer membrane. So remember, the envelope is, you know, the cytoplasmic membrane, the wall, and if you have that outer membrane as well. that whole thing is going to be the envelope of the bacteria. So endotoxin is basically lipopolysaccharide, which is a molecule on the outside of that outer membrane. When you guys see for gram-positive, thick cell wall plus teichoic and lipoteichoic, so those are kind of come together, then I want you guys to think if it has an outer membrane, gram-negative, and endotoxin should come with that. And just think that's the same thing as LPS, which is lipopolysaccharide. It's just a marker on the surface of the cell that our cells see as foreign, which we'll get into when we get into immunity. So I don't want to talk about it too much. So that reminded me about sequence of questions on the exam. So it's set up to be done in an order, and it's set up in that order. But the problem is there are occasionally questions that depend on the answer from the question before. I tried to eliminate them as much as I could and like reword it so that even if you didn't get that one, you can still answer the next one. But there are, I don't know how many, but there has to be at least one or two on there that are going to be those. So I know that's a cheap move.
Does it repeat the question again on the next one, or you just have to
You can go back. So
okay.
I do have it set up where you can always go back to any of your questions at any point. It's one question at a time, but you can backtrack. So as long as you get it done within that time frame, you're good. Okay, next thing. What type of coating is a capsule or slime layer? Right, so the correct answer for this one will technically be glycocalyx, because the other ones, protective and defensive, that's just depends on, I guess, how it's being used at the moment, but it's also used for adhesion, those things. So it's not just those, so don't forget. And obviously it's not just carbohydrate, because sometimes there's protein in there. So good job with that part. Let's go on next thing. All right, that's it for chapter 4. I'd be aware of what peptidoglycan is. Remember, it's repeating sugar molecules, NAM and NAG. I am not going to ask you to draw it out or tell me about the chemical composition of it, but I would be aware that they are repeating sugar molecules. Other kinds of bacterial cell envelopes. So we have gram-positive, we have gram-negative, we have the ones that do not have a wall. So those are the mycoplasma. And then we have the ones that are special with mycolic acid. And does anybody know what those ones are? Right, so mycobacterium, right? So yes, anything mycobacterium, whatever. Quorum sensing is whenever we have buildup of bacteria on top of themselves and they're living like that, like plaque on your teeth, literally. So they can communicate with each other via chemical signals, letting them know that, this whole mess is doing pretty good right now, or if they want to quit. So that's quorum sensing. And then we have endospores. That's, I feel like, pretty straightforward one at this point, again, because we did it in the lab, but remember they are protective structures. They're formed in harsh conditions to protect that genome until good conditions arrive again. And I'd be aware of that kind of cycle of how they're made and the terms involved there, right? Like vegetative and all that. All right, eukaryotes now. Which of the following is not an organelle commonly found in eukaryotes? Right, so most eukaryotes are going to have all three of mitochondria, endoplasmic reticulum, and Golgi apparatus. The nucleoid is a region in bacteria where they maintain their genome, essentially their chromosome. So that's not even technically an organelle at all. Remember, your mitochondria makes energy. Endoplasmic reticulum is where we have molecule manufacturing, like protein manufacturing and all that. And then we have the Golgi apparatus, which modifies and packages that stuff. Right, true or false? Fungi are never pathogens. That should be an easy one. Man. All right, so that's gonna be false. We do have fungi that can be pathogens. So that includes fungi that can cause fungal meningitis, or athlete's foot, or ringworm, that's fungal as well. There's others, of course. All right, moving on. So what structure in bacteria can protozoa trophozoites be compared to? So the analogy, I guess. I know. All right, so the answer is going to be vegetative cells. So the trophozoites are the free living ones that are swimming around, living their lives. That's similar to the vegetative cells for bacteria. So this is a multi-select. What is a cestode? I would not go by the picture. If you guys are looking at the picture, that's not it. Okay, so our correct answers are going to be tapeworm, helminth, flatworm. So remember that the helminths, we can break it down into platyhelminthes and nematodes. Nematodes are the roundworms. So this is a flatworm. I mean, if you know about the structure of it, you can tell just by looking at it, right? And then in the platyhelminthes, the flat ones, we have the cestodes and the trematodes. And the trematodes are the flukes. So that's just one that you kind of have to know. But I'd draw out one of those little like branching off things. I'm sure there's a word for that. That makes more sense than branching off things. So for the eukaryotes, you will want to understand what the organelles are in a eukaryote and what their functions are. I would be aware of the fact and remind yourselves that prokaryotes, like bacteria, do not have any organelles at all. So if you ever have a question on the test that says something about bacterial organelles, they do not have one, okay? But eukaryotes do, and I'd be aware of what they have. So fungi, cell structure, and roles in the biosystem. Again, people don't know a lot about the fungi, so I just go back and review the basics over those. The algae and their roles in the ecosystem. Remember that algae are protista, and they are the #1 producers of oxygen on the planet. I think it's something like 70% of our oxygen in the atmosphere is made by the algae. Characterizing protozoa by their methods of motility. In the chapter, it does like classify the different protozoa based on if it has, the cilia, or if it has the amoeboid movement, the pseudopods, or if it has a flagella, or if it doesn't move. So those are the 4 ones that they could be. And then characteristics of the helminths, which we kind of just went over the basics of that. Okay, next. We know those are the parasitic worms. That's the helminths. Okay, chapter 6, these are the viruses. Again, multi-select. Pick all of the structures that comprise the absolute bare minimum for a basic functional virus. The bare minimum. All right, so our answers are capsid and nucleic acid. That's it. That's the bare minimum of what you need for a virus. And I did in the question technically say naked virus, but also kind of gives it away too, if anybody caught that. But yes, so some viruses do have an envelope, but all viruses have to at least have capsid and nucleic acid required for all of them. All right, next thing. What is the purpose of the viral spikes? So I'll say that this question is the kind of question that I feel like the test has a lot of, which is I'm giving you choices that you really actually have to understand this concept in order to get it, right? So the purpose of viral spikes, they're on the outside of that virus to help it get in to the host cells. That's what they're for. But yeah, basically that's what they're for. Moving on to the next. Oh geez, that didn't mean any of that. Okay, what is lysogenic conversion? So lysogenic conversion, this is whenever we have the phage, the bacteriophage is a temperate phage, which is what the term we refer to it as, and it inserts its genome into the bacterial genome, and it becomes a prophage at that point, and also becomes lysogenic at that point. So we could just stop there. But when we have lysogenic conversion, what we really have going on is when we insert that, we have also introduced new genes into that bacterium that are typically virulence factors, things that help them survive better. This is the case with Corynebacterium diphtheriae, which is diphtheria. Its toxin comes from lysogenic conversion. So the whole reason that it is bad is that. So it's kind of neat to think about that kind of relies on that. So that's what's going on there. Okay. Next thing. What special superpower does reverse transcriptase impart to HIV? I have a lot of questions like this on the test, by the way. I don't mean like content wise, but I mean like I gave away kind of the answer in here if you understand the words that are on the screen. So what special superpower does reverse transcriptase impart to HIV? If you know what transcription is, you should be able to answer this question. Transcription is DNA to RNA, and translation is RNA to protein, right? So if we're talking about reverse transcriptase, which would be DNA to RNA, do it in reverse, RNA back into DNA. So it tells you the answer kind of in the word. And it almost is like, you can get there two ways. You can either just know what reverse transcriptase is, or you can look at the words and understand what it would mean in that context. So yeah, so there's kind of a lot of questions like this on the test. I don't know. So be sure you read through those questions because it could actually be a good thing. It could give you a hint about something that you do understand better. So chapter 6, bacteriophages. Remember, those are viruses that infect bacteria. We'll forget that somehow. Latency versus lysogeny versus lytic phase. We're talking about bacteria. We're talking about lysogeny and the lytic phase. So it's just whenever the virus is active and bursting out of cells, that's the lytic phase. Lysogeny is whenever it's the prophage incorporated into bacteria. With latency for humans or males, eukaryotes, what that is essentially going to be is whenever that virus goes into hiding somehow. Often that can mean that it's inserting its genome as a provirus into the host genome, but it doesn't have to mean that. They can hide out in other ways as well. So that latency is whenever they hide out and they are no symptoms for a while, and then they come out due to stress or something like that. I always use herpes as an example of this because I think it's a pretty good comparison. So oncoviruses, cancer causing. And just think of oncology. And then prions, those are proteinaceous infectious particles. They're literally just proteins that are misfolded. They stick together because of that. And when they stick together like that, that can poke holes in your brain and then you die. I mean, that's the less glamorous aspect of it. You can be harboring this for decades Once you start showing symptoms, typically death will occur within a year. So nothing you can do about these, but they're super rare. So don't freak out about them or anything. They're super rare. Okay, chapter 8, microbial metabolism. I love metabolism, so.
I want your review, but you need to, this is my favorite chapter. I was like,
No. So what type of reactions involve breaking chemical bonds to harness energy? All right, so that's me. Catabolism. Remember, cats destroy everything. That's how I remember it. All right, so next question. What is non-competitive inhibition? I feel like there was a lot to cover for this chapter that I have 4 questions to work with. So. Okay, so non-competitive inhibition. We will have I'm sorry, I had to like a word limit on these. So some of them are not worded as much as I would like for them to be explained well. So basically you're making you're taking A as your substrate, your enzyme binds with A in that actual active site where it's going to work on A and turn it into B. All right, we build up enough B comes and binds somewhere else on that protein called the allosteric site or the regulatory site, both are the same thing, and causes a protein shape change when it binds at that back site or whatever. Now it can't bind onto A anymore. So that's how that's going to work. That's non-competitive because the only difference between competitive and non-competitive, the only difference is that competitive, you're binding in the active site. It's probably the better way to remember it. It doesn't have to be that confusing. Okay. How does ATP store energy? Right. It's going to be all about the phosphates here. So the phosphates will interact with the enzymes. What?
I thought it was.
Yeah, I guess that's not, yeah, you guys are probably right. I'm reading it and I'm like, that's not right. So you guys guessed this one, right? Absorption electrons. That's not right at all. So it's the blue one. So that one, it's the blue one. So phosphates will repel each other and the energy will be released when the bonds break. So think of it this way. We have a whole bunch of north magnets. They're pushing against each other, but we force them to actually be bonded together. So there's all that potential energy that's building up. Potential energy is like if I were to ask you to hold, I don't know, imagine that this way is like, 40 pounds or something. And I ask you to hold it up like this. What's going to happen after your hand or your arm gets tired? It's going to fall, right? It has to. Why? The potential energy that is built up from it being up here has to be released. That's what's going on with those phosphates. They're pushing each other apart so much that they want to be broken apart. So when you break one of them off, it releases a ton of energy and can power the other stuff. So that's how ATP will work. Does that make sense? Because they're negative. So I don't know if you guys remember, but phosphates is like a P with an O's all around it. And we know oxygens are negative. They want to steal electrons. So we know that has to be a very negative, molecule group, functional group, right? That's the right way to refer to it. Okay, cool. All right. Which molecule serves as the final electron acceptor in aerobic respiration? All right, so the answer is going to be oxygen. Talk about that a little bit more when we get to the end of it. Okay, that is the end of it. So, okay, enzyme structures, terms, and regulation. It's hard to break it down any more than that, but what an enzyme is, right? A holoenzyme, apoenzyme, coenzyme, all of those terms that are associated with it. and what they mean and how they be regulated, competitive versus non-competitive. Right, so constitutive enzymes, enzyme classes and redox reactions. So there's those 6 main enzyme classes: the oxidoreductases, the liases, the ligases, the hydrolases, the isomerases, and I always forget another one. Did I say oxidoreductase? Huh?
Never mind.
Oh, yeah, you're fine. But anyways, I know those classes and basic concept of what they are. I don't need you to know exact chemical breakdown of what they do, but just idea of what they are and why they're important. Remember, redox reactions are any time that we have exchange of electrons at some point. That'll be included with the redox reaction. You'll want to know what reduction is and what oxidation is. I'm sure you're sick of hearing about it, but yeah, I would be aware of those. So I remember, as Leo says, GR, lose electrons oxidized, gain electrons reduced, but there's also oil rig that some people like to use. Oxidized is loss, reduction is gain. So I just think of that one because that's not one I always use, but whatever works for you. Okay, ATP, we did talk about the phosphates. I feel like that's the most important aspect about ATP. Be aware of what it is made of. It has 3 major components. So it has that adenine nitrogenous base, 5-carbon sugar. Does anybody know what 5-carbon sugar is in there? It's okay if you don't, I'll tell you. It's ribose, okay? Just like RNA has. So adenine is our nitrogenous base, ribose, our 5-carbon sugar, and then 3 phosphate groups. So yeah, it's basically a nucleotide with extra phosphate. If that doesn't make sense to you, then you might want to go look up nucleotides, which is the next chapter. All right, aerobic respiration, anaerobic respiration, fermentation. Anaerobic respiration, really the only thing that I want you guys to know about it is that it's what anaerobes do, right? Proper anaerobes. So I'm not talking about facultative anaerobes, I'm talking about actual anaerobes. They do not use oxygen. They might even be incapable of using it. So those are the anaerobes. They will do anaerobic respiration, and they use anything other than, not any random molecule, but a wide array of things, but it's never oxygen at the end of their electron transport chain as their electron acceptor. Aerobic respiration, which we just talked about, oxygen will have that. That's what makes it aerobic. So we have that final electron acceptor is oxygen. And then fermentation, that's just what facultative anaerobes do when there's no oxygen. With the breakdown of how these all work, I would know which ones have glycolysis, Krebs cycle, electron transport chain. Those are the big picture things, right? So fermentation is just glycolysis. And yes, we get pyruvic acid, which is the most important thing we're getting out of glycolysis. We get it there in fermentation, and we have to alter it to make the NAD, NADH thing take care of itself again. That's where we get byproducts of the acids and the alcohols and carbon dioxide with that. So that's how fermentation works. That's pretty straightforward there. And then aerobic respiration, which is basically the same thing as anaerobic, or it can be, but just with a different electron acceptor at the end. Aerobic respiration, glycolysis. Glycolysis breakdown of glucose, a 6-carbon sugar, that's the important part. Glucose, 6-carbon sugar. We break it into 2 3-carbon things, 2 pyruvic acids. That's basically the end result of it. So a pyruvic acid will eventually work its way through the Krebs cycle. So you can do it twice with each glucose. So that's important as well. Then in aerobic respiration, we have a pyruvic acid, and it's going to get converted into acetyl-CoA. That's like just an intermediate thing that's happening. And that does give off carbon dioxide at that point, because it had to do with a carbon exchange off of there. But the big deal that's going on is that then we go into the Krebs cycle. And Krebs cycle is basically just shoveling around electrons and carbons. So we can move electrons onto electron carriers like NAD, like FAD, to turn them into NADH and FADH2, right? So we make a whole bunch of that in the Krebs cycle. As we're going through that and knocking off molecules, we lose carbon dioxide in the process as well. So, so we've made NADH and FADH2, right? That's what we did in Krebs. That was the whole point of Krebs. Glycolysis, making pyruvic acid. Krebs, making our electron carriers. Now we're going to enter into the electron transport chain where we're going to let loose those electrons. They're going to shuttle their way across those cytochromes, which are those proteins in the chain. And as they're doing that, we're going to shuttle those protons that don't have the electrons associated with them anymore, the H's, H+. We're going to shuttle those outside of the cell, build up a gradient of them. They want to come back in because there's too many of them outside. The only way they can come back in is ATP synthase. And when they do move through ATP synthase, it powers ATP manufacturing. So that's what powers the actual putting of a phosphate on. You would need energy to want to put two north magnets together. So that's how that's all cycled. Now, the problem is we've done that. We've made our ATP and we think we're done, but we're not, because we have to keep doing that. We can't build up every waste product. We need to get rid of it. So we have what's left at the end of it. We have the electrons that shuttle through. We have the protons that we pump back in. And then we have hydrogen. Oh, we did the protons already. Yeah. Protons and then, yeah, okay, whatever. So the electrons and the protons. So what we do to help neutralize the excess of these things, we put those electrons back with the hydrogen, but they have to be bound to something when we do that. So we take half of the oxygen atom and do that. So now we make water. That's why we need to breathe in oxygen. It's the only reason why you need to breathe in oxygen. And it's also when we went through Krebs and released carbon dioxide, that's the only reason you exhale carbon dioxide. And people think that the oxygen from that, when you breathe in, is the same oxygen as the oxygen molecules that are in the carbon dioxide you're breathing out. Nowhere near the same molecules, right? Because the oxygens that you're breathing out came from the molecules that went through the Krebs cycle. The oxygen that you had breathed in is going straight at the electron acceptor. They don't touch each other. So a lot of people don't realize that, but now you guys do. I think that's why I think it's the coolest thing. And that's why I talk about it a lot, because I feel like it's a whole process that is worth understanding, but a lot of people don't. So, so I hope that kind of was a run through of that. Right, but I teach this all the time. So of course, to me, this is like straightforward. And then photosynthesis. Photosynthesis is kind of what's going to be allowing us to have that oxygen that we breathe. I would definitely be aware of this. So photosynthesis, we know light is going to provide the energy for all of this. Light excites the pigments. When we know that, that allows for the same thing to happen, proton gradient. Proton gradient, just like we had in our thing. But it's only going to allow us to make a little bit of ATP. That ATP that's made from that light reaction is going to go straight into powering the dark reactions or the Calvin cycle. And that's basically using that ATP and carbon dioxide from the air to make glucose. And that's what plants do. And then what do they do with that glucose? They use it in aerobic respiration that we just talked about. And it's going to give them a ton of energy breaking that down. And they don't need as much necessarily as we do. We're running around and doing stuff. They're just sitting there and growing. So that's why they're actually going to be giving off more oxygen than they're going to need for the energy. Cool. I think that's pretty cool. Don't forget that plants do need to go through aerobic respiration. If they didn't, they wouldn't have mitochondria in their cells. It's all that mitochondria does. All right, genetics. This is worse, I feel like, for a lot of you. So we'll do genetics. Let's run through this. We only have 4 questions. So true or false, bacterial transcription takes place in the nucleus. This is a trick question, by the way. Yeah. All right, so the answer is going to be false for this one. Bacterial transcription takes place in the nucleus. Number 1, there's 2 things here that point to false. Bacterial transcription doesn't take place in the nucleus because there's no nucleus. So it can't. If it had one, it's the nucleoid region, right? So that's where this is going to happen. But that's important to understand, because if you have bacterial transcription and it's just hanging out in the cytoplasm, what else can you do at the same time as you're making your mRNA, your transcript? You can also pop one of those ribosomes right on there as you're making the same transcript and start making the protein immediately before you have even made your transcript whole. Like while you're actually making the mRNA, you can start making the protein. Only bacteria can do that, which is nuts. They call that co-transcriptional translation. All right. All right, the reason Okazaki fragments form during DNA replication is due to, why do we have them? Right, so our answer is going to be the strands are anti-parallel, and that polymerase is moving in only one direction. So because they're oriented in different directions, we can't just go straight in to where we're opening up and unzipping our DNA on both sides. We can only go one way on one strand and the other way on the other strand. However, the strands themselves being anti-parallel, facing opposite ways, we can't just go straight down. Polymerase only moves in 5' to 3', so front to end. It can only go in that direction. And so what it has to do is, as it opens up and we get the big opening up, then we'll go on and make a little bit of what's given to us, and then it'll open up more, and we'll make another little bit of what's given to us, and they keep doing that as it opens. So we have these fragments that are forming, and those are Okazaki fragments. That's what those are. So that's on the lagging strand during replication. All right, next thing. All right, which of the following is not a means of horizontal gene transfer by bacteria? This concept is one that I like to test over a lot, FYI. All right, so translation is our answer. Remember, transduction is whenever we're going to be moving genes into a bacteria using a phage, a virus, okay? Transformation is whenever the bacteria takes up DNA that's in an environment. It's often what we would use for genetic engineering. That's what I'm looking for. All right, and then conjugation is whenever we have the two bacteria connecting to one another via the sex pilus, and then they'll transfer that DNA between each other. Translation is whenever we take our messenger RNA and turn it into protein. So it's a whole different thing. All right. If a single base pair is deleted entirely, what type of mutation will this cause, if any? Deleted one base pair, what would it do? So the majority did get this. This is a frame shift mutation. It's just like if you have a word or a whole book, right? And you had to keep reading the book. If I delete even like one letter, well, it shifted the whole letter count over by one, right? It's the same thing here. If we're going by threes and sets of threes, then basically like if we shift everything over by one letter, by removing one, then that's going to read a completely different thing as we're moving down the line. So that's a frame shift mutation. That's shifting the frame that we would be reading and causing completely different amino acids as a result. Remember, those amino acids are really important for the structure and therefore function of the protein. So chapter 9 obviously didn't cover a lot in these questions. Know what a genome is versus a chromosome versus genes. Remember, the genome is every genetic thing in that organism, including plasmids and crap like that, even extra chromosomal, like we say, DNA. Chromosome is what's actually going to be that unit that contains the genetic material of that organism. Specifically, some organisms have more than one, like eukaryotes typically have more than one linear, whereas bacteria usually have one circular one. It can be different though. And then we have genes, and those are little packets of information that code for usually a functional product of some kind, and that's what we would call a gene. Genotype versus phenotype. The genotype would be like if you got genes from your mom that gave you brown eyes and genes from your dad that gave you blue eyes, but you have brown eyes. You don't have like one blue eye and one brown eye, right? So that's your phenotype is that one color, but your genotype is you have the genes for both of them. All right, characteristics of DNA and RNA. I mean, the DNA is going to be double-stranded. We're going to have deoxyribose as our sugar instead of ribose for RNA. We use ATCG for DNA, whereas AUCG for RNA. And the U is uracil, T is thymine. Replication, we kind of went over that a little bit, but you would want to know what the details involved that. Transcription, oh my gosh, introns and exons. Transcription, remember, that's going to be making that mRNA. So the way that I like to describe it is, you have the Smithsonian Institution, and that's this massive book on how to build all these different kinds of houses, but you only want to build this one house, and that's the only place you can find that information. So you go to the Smithsonian, you write down or make a copy of that one house, and you take it home, and then you're going to start translating that information on those instructions into a house by building it, right? So that's exactly how transcription and translation works. Transcription is writing down that copy of that one thing. So making a copy of like mRNA. So we have mRNA, ribosomes will read it and spit out protein. Okay, great. That's great. That's how that works. Fine. Eukaryotes have this crap in them called introns and exons. In their actual mRNA transcripts, they'll have to cut out the introns, which don't code for anything. And there's reasons for all this. We're definitely not going to get into that. And we're going to seal together the exons. Only eukaryotes do this. So if you put straight up like your genetic information into a bacteria and try to force it to make your genes, you're going to have introns in there. And they can't deal with that. They don't have introns in their cells. So they wouldn't be able to make your proper protein. So we have to actually take our edited version of our mRNA that we would actually use and turn it back into DNA in order to put it into the bacteria when we're engineering. So that's important to remember. Triplets, codons, anticodons, amino acids, those are all linked together. Triplets, the 3 nucleotides on the DNA. Codon, the corresponding 3 nucleotides on the messenger RNA. Anticodons, those 3 nucleotides that are the match to the codon, but now this is on the transfer RNA. That transfer RNA, if it matches up the anticodon to the codon, is attached to a specific amino acid, and those are get linked together to make protein. That's how translation works. That's basically translation. I don't think you really need to talk about that more. And then we have the operons. I hate them. I hate the operons. So the operons, you have basically repressible operons and inducible operons. The last operon would be an inducible operon. We don't usually make the genes associated with breaking down not us, bacteria. I want to be clear about that. Bacteria do not usually make the genes associated with breaking down lactose until lactose appears, and it's there. Then when lactose does appear, it binds with the repressor protein, which was sitting there blocking, making the genes associated with breaking it down, causing it to change its shape and move away, and now we can make the genes when lactose is present. Eventually, that's not a permanent relationship, so it'll let loose, and if there's not enough lactose keeping it off, then it'll eventually bind back on and stop it. So that is inducible. We induced it with the lactose. It wasn't already on. Repressible, it was already on. It's something that it does. So that way the bacteria are constantly making, let's say, an amino acid arginine. And basically what it'll do is, as it's making that amino acid, it will build up an excess of it. And eventually, whenever that excess of it is built up, it binds to a repressor protein as well. But this time, the repressor protein will go bind on and block things when that happens. So the product there is telling you we already have enough of this, so you can turn it off. So that's how repressible ones work. Cool. I know that was a lot. Chapter 9. All right, chapter 7. This one's not so bad. This is microbial nutrition. All right, regarding the elements in the cells, chunks, what is S? I just pick random ones. It'll like suggest things.
Get good here.
Yes,
Yeah.
yeah.
Some of them, yeah, some of them lazy on. So regarding the elements, what's the S? This is going to be sulfur. Okay, good job, guys. Good job, I'm proud. Yeah, multi-select. So what are symbiotic relationships? You can choose more than one. Doesn't mean you have to choose more than one, I'm just saying. All right, so our answer is going to be when two organisms live closely and at least one is dependent on it. And whenever we have both organisms benefit from that close relationship, that also, I mean, is a type of symbiosis, I guess, right? So, and then also so is when organisms would die without the other, and each helps each other. You can tell I didn't have I had word count that I had to make. But basically, yeah, if you have any sort of a relationship where one organism depends on the other one, whether it ends up being harmful, helpful for both, or only helpful for one of them, that's symbiosis. But if they rely, so at least one of them has to have it, that is it. So these qualify.
I have a question.
Yeah.
Wouldn't the red one be parasitic? Is that not?
It is parasitic. I'm trying to think of why I didn't select that. There might have been a reason, or maybe I just like, I don't know. Yeah, you would think that would be. I don't know. It should have been all of them probably. So that one I guess got left out for some reason. Sometimes when I go to check them, I'm not very good. It will be like 2 o'clock in the morning. I'm not gonna lie. So that's why these ones aren't for credit. Okay. Multi-select. Which of the following is a primary producer? Okay, so primary producer, that's going to just be the algae. So none of the other ones were. If you have to, if you don't depend on, so you'll be a weird thing if you get your energy from not sunlight and your primary producer. It could be certain kinds of bacteria or something can do that. But typically, most of the time, your primary producers are going to be your photosynthetic organ. Typically. I will not ever introduce a scenario where it's not. I'll tell you that, okay? Because I don't know enough about the other ones. But yeah, so all of these guys depend on other organisms having made carbon molecules, so organic molecules, right? Algae and other photosynthetic, they make their own organic molecules from the carbon dioxide in the atmosphere. Okay. All right, in a bacterial growth curve, what is happening in the stationary phase? All right, so yes, that's correct. Growth slows and equals the death rate while nutrients start to run out. So we're just kind of plateau, right? All right, chapter 7. Terms for where organisms get their carbon and energy. So that's, we're talking about heterotroph, chemotroph, autotroph, all of those. Be familiar with those terms. Energy flow in the food pyramid, again, always going to be at the bottom. We're going to have the primary producers moving up, and there's less energy the higher up you go. Temperature preferences, that's going to be like psychrophiles, like to live in the cold. Psychotrophs can deal with the cold. Mesophiles, those are at room temperature and pathogen temperature. Pathogens are mesophiles. Thermophiles like higher temperature, and then the extreme thermophiles, particularly the archaea, can live in like boiling water. Okay, so those are our temperature preferences. Diffusion versus osmosis. Well, diffusion is movement of molecules from an area of high concentration to low concentration, just like if you remove a net and let the bouncy balls roll out. So that's diffusion, and osmosis is diffusion of water molecules specifically. And then be familiar with the concept of the different kinds of membrane transport. So we could have just diffusion, where we only really see in our membranes normal diffusion is osmosis of water molecules. And some other small molecules can too, just get across the membrane without any help. If they're going with the gradient, like from high to low, but they can't get across the membrane by themselves, they just need a channel, and then they can just go through the channel if it's there. That is facilitated diffusion. There's no added energy there. If it requires energy for some reason, either you want to move faster, so you put energy in to go faster with the gradient, or you want to go against the gradient for some reason, which of course you need energy if you're going to fight that, like fighting a current, literally. So that's active transport. So you need energy. Neat. We're getting there. I clicked it, right? Didn't I? No, I didn't. Chapter 10, this is genetic engineering. Which of the following is not a major step in PCR? All right, so this is going to be termination. There's no termination step for it. They'll just keep going on. Denaturation, anneal, elongation. Denaturation, anneal, elongation. Another thing that I would be aware of for the exam. Definitely on, I think it's on the lab exam, but still. PCR. Anytime that you have DNA and you separate it and you make a new one out of it, so now you've got two, right? If you want to go another round, how many would you have after that? 4, because each one will make 2, right? You need to be able to tell me how many, copies you would have at the end of X amount of rounds. So if you need to draw it out, that's fine, whatever. But yeah, so be prepared for that. Like if you go through 4 rounds of PCR, how many would you have at the end? Double-stranded products. Make sense? That's going to be on one of them. I know for sure. To me, they've kind of run together in my mind, but I know that's on there. What are restriction endonucleases? Yep, so all of these are correct. They are enzymes that bacteria produce to get rid of foreign DNA that cut at specific palindromic sequences, leaving sticky ends. All right, plasmids contain genes that are essential for bacterial survival. True or false? All right, so that's going to be false. Plasmids can contain genes that can help, increase the likelihood of survival, but they shouldn't be containing any genes that actually you need it to survive. That's contained within your genome. The plasmids are separate from it. So you should be able to survive without a plasmid if you're a bacterium. All right. Next thing, why can't eukaryotic genes be placed directly into bacteria to create the target protein? Let's see if you guys are paying attention. All right, great job. I wasn't expecting that. I thought it would be like 50-50 introns and exons. Pretty good job. So it is introns. Need to get those suckers out. All right, chapter 10, you're going to want to look over the steps in PCR, the ingredients that you would be absolutely necessary in PCR, and the theory of how it works. Gel electrophoresis concepts. Again, this was stuff that you guys did in your lab stuff online, but know, where the larger molecules would migrate versus the smaller molecules, and referring to DNA size in terms of the number of base pairs. Transgenic organisms and GMOs, those are the same thing. Number one, be aware of that. Number two, anytime that you put foreign DNA from one organism into another organism, that is a GMO or a transgenic organism, including whenever we have bacteria producing insulin. And then gene therapy is whenever we're trying to introduce working healthy genes into somebody who does not have a healthy version of it. They're lacking in production of some sort of protein. We try to put a new gene into them and make it a permanent part of their actual body. Yeah, I don't know. But yeah, well, you can use viruses to do that. And we usually mix it with their bone marrow and infuse it back into them and hope that it takes. There are currently, I think, I'm wrong, it could be 18, but I think it's 13 diseases that are approved for treatment with gene therapy right now. Yeah, you're fine, I understand. Chapters 24 and 25, this is environmental and applied microbiology. This question is mostly about water treatment. So in the carbon cycle, what roles do photosynthetic organisms play? Yeah, so the answer is going to be all of these. So it actually does do all of these. We're going to fix the carbon dioxide into organic molecules that can be used by the consumers. Then we also have, if the plants die, the saprobes are going to break them down and use those organic molecules as well. And then organic molecules that are created in that photosynthesis will also be used in aerobic respiration, which I kind of talked about earlier. So cool. Next. When treating water for drinking, why is water aerated? The answer is going to be it kills obligate anaerobes, because remember, they don't use oxygen as their final electron acceptor, so they don't have the means typically to protect themselves from toxic oxygen byproducts. All right, next thing, during wastewater treatment, what is done with sludge? Yep. So if you have sludge, it's just the part that isn't truly liquid, but it's kind of like mud whenever you mix it up. So basically that stuff, we can get bacteria to break it down further so we can save even more of the water. So not too much of it's going to waste. And make it usable products so that we can, not just have poop basically. So what are coliforms? That's okay. So the answer for this one, these are bacteria that if they are there in the water sample, it indicates that there could be pathogenic organisms either there currently, or they could, live in that situation. So you want to, get that situation taken care of. All right, chapter 24 and 25. Oh, I know about biomes, what they are, and examples maybe. I don't know, like the tropical rainforest is a biome, for example. Element cycles. We talked a little bit about carbon dioxide. I'd be a little bit aware of the nitrogen cycle as well. Energy flow in the food pyramid. Again, we talked about that in the other slide, but it was brought up again in this. Potable water. Potable water is an important concept. Geez. That basically means that the water does not have any pathogens in it. Potable comes from the Latin potare, which means like drinkable, I guess. But anyways, water treatment and wastewater treatment. I go through those cycles, just get the gist of the concepts. Microbes in food, of course. So like using it for bread, wine, pickling things, beer, and then food safety concepts. And one of the ones is just to remind you guys that putting something in the freezer or keeping it in the fridge doesn't actually kill bacteria typically. Very rarely does it kill bacteria even when you freeze it. All right, chapter 11, moving on to microbial control. So these are just mostly going to be memorization. So traditional autoclaving will destroy all microbes, even prions. So the answer for this one is going to be false. Prions can survive autoclaving because they're not organisms, so they're just protein. Yeah, so you have to autoclave them, you can, but you have to do it at 135 degrees Celsius, which is the maximum that autoclaves can operate at, and you don't usually use them at that temperature. Unusual. Typically, you also want to treat them with sodium hydroxide to denature the proteins too. They're hard to get rid of. So, what is the term for removal of microbes from animate or living surfaces? All right, this one is going to be asepsis. Again, like a lot of these are just memorization terms, so hang in there. All right, true or false? Ionizing radiation causes the expulsion of orbital electrons. All right, so that is true. And that little picture that was up, was a picture of an old amusement park that was in Pripyat in Ukraine, which is where Chernobyl happened. That's Chernobyl. So I think that was pretty cool. It's the creepiest thing you'll ever see, though. They have like abandoned, amusement park and everything. They had just built a whole bunch of new apartments too. Which of the following chemicals of microbial control is capable of sterilization when used properly? All right, so that is going to be the halogens. Like I said, this is more or less memorization. So I could go through the chemistry of all of this, but it really is just memorization. All right.
Good. You need a break in there.
All right, so other things for chapter 11. You want to know the difference between microbicidal and microbistatic, of course. Which methods of microbe removal are appropriate for what? Whether you're, you can't autoclave a patient bed, right? So that's what we're talking about there. How each method works. Does it affect the cell wall, the membrane, the proteins, or the nucleic acids? And then which ones are capable of sterilizing if used properly? All right, next one. Is that it? No, we have still 13 and 17. That's right. Oh, geez. Okay. All right, 13. What is the correct term for pathogens which originated from outside of your body? If it's from outside your body, that's going to be exogenous. Okay. All right, multi-selection, guys. So select all of the examples of indirect transmission. All right, so the answer is going to be I sneeze on a keyboard, you use it afterwards, after I'm gone, and then I catch my Ebola. So that is going to be an example of a fomite. The keyboard would be the fomite, right? And then the other one is I use the toilet, I don't wash my hands, and I make you a meal, and then you catch my Ebola when you eat it. Now, technically there, the meal could be considered a fomite because it is, you know, the vector for transmission. So you could look at it this way, but yeah, that was indirect, right? I didn't transmit it directly to them. Yeah, good job. You guys did really well on that. Okay, what is the correct term for lasting effects even after clearing an illness? No. No, there's only one. There'll only be one answer for all of those questions. All right, so the correct term for lasting effects is going to be sequelae. Good job, guys. True or false? Okay, incidence refers to the overall frequency of an illness within a population. It's supposed to be overall frequency. That was supposed to be true. That's on me. Yeah, I'm like rereading it. I'm like, why would I put false? I think I changed my mind on how I was going to word it, and then I never changed the answer. I was like, I had too many that were false. So I thought I needed a true, and then I never put it as true. So that's my fault. That is incidence is overall, right? If you're talking about prevalence, no, that was right. Now I've got to think about that again because now I'm thinking prevalence is overall. Incidence is over a certain period of time. So that was correct. Yes. So if it's over a specific amount of time, like how many cases occurred in the last year, oh, that tripped me up so bad. Put me out of all these chapters, man. I know how you guys feel. Anyways, prevalence, that's going to be overall over time, like what you would expect the frequency to be. Incidence is going to be referring to like over a year, how many cases of COVID there were and how frequent it was in that year. That is also the same thing as the morbidity rate. Okay, cool. Oh, sorry about that one. Got confused for half a second. All right, chapter 13. Remember, terms, pathogens and disease. Pathogen, again, would be any organism that could create a disease-like symptom in your body. And disease is any deviation from your normal health. Microbial antagonism, whenever like we have microbes taking up all the stadium seeds, right? And then the bad guys can't sit down. And also taking away all the nutrients from the bad guys so they can't grow. Anytime microbes are staving off other microbes. Antiphagocytic factors and virulence factors. Antiphagocytic factors are anything that prevents phagocytosis from being successful. Virulence factors are anything that helps the organism establish infection and pathogenicity. Steps of infection, it's literally just going through like, you know, portal of entry and, you know, adhesion and all that stuff. Signs, symptoms, and syndromes. Signs are going to be measurable. Symptoms are going to be reported. And then syndromes are basically medical conditions that consist of a set type of signs and symptoms that will be associated with it, but can be caused by a lot of things. That's a wordy way of saying something like meningitis, which can be caused by a lot of things, but they all have the same kind of symptoms. Or pneumonia, same kind of symptoms, all can be caused by a lot of different things. Or the common cold, caused by 200 different viruses. So, but it all has similar things. So that's a syndrome. All right, so signs are measurable. Remember that. Symptoms like pain, you can't measure somebody's pain. Reservoirs, carriers, and source. Reservoirs are things that are maintain the existence of the pathogen in a given area, whereas carriers might have been infected or have the pathogen growing within them, may or may not know about it, may or may not have symptoms, but they are the one that's going to go give it to other people by exposing other people to it, but they're not the reservoir, not where the illness is maintained outside of that. The source is going to be if you have an outbreak, that's where it came from. Like either it could be a person, which would be point source, so be one person led to the spread of all this stuff, or it could be I can't remember. No, I feel bad for that. The other one was where over time you can have a population exposed to a situation that would expose them to this illness, because maybe they all go to the same drinking spot or whatever, and they all end up sick over time, but not from one person in one event. Common. All right, zoonosis. So that's anything that's transmitted by animals, typically found in animals, and then we can get it from them, and we are the dead end hosts in that case, typically. And we can't transmit it or maintain it in a human population after that. The transmission, and then epidemiology. So transmission, that's really referring to the different ways that it can be transmitted, like we were talking about with direct and indirect and fomites and all that. Be aware of, like, when you sneeze versus, those droplet nuclei that dry up, all that. And then fecal oral, of course. Then we have epidemiology terms. We talked a little bit about that with the reservoirs, carriers, and the source stuff, but this is where, prevalence versus incident, morbidity versus mortality, and all that would come in. Okay. Last one for this chapter, the chapter thing. Diagnostics. So chapter 17, I multi-select. Which of the following can be used for identification of an unknown bacteria? Select all that apply. So it can be all of them. All right, what is a Western blot? Good job. Selectivity means that all positives will be found at the risk of false positives. This one's false. Yeah, I figured it was going to be split. So that would be sensitivity. That's the definition kind of what you can get with a super sensitive test. So even if there's minute amounts of the antigen or whatever, a super sensitive test will detect those minute amounts. However, they might also bind with things that aren't that positive because they're trying so hard to bind with a potential positive. Next, the selectivity. That one is going to only bind to that antigen and nothing else, not even things that look even remotely like it. But then you're risking false negative with the selectivity because you might not bind with every case of that positive antigen. Okay? Because you're being too selective. All right. Obviously, you want both, right? All right, a direct ELISA is testing whether or not the patient has the antigen. Pretty good job. So yeah. All right, chapter 17, a diagnostic process, like basically where would you start and where would you move on next? Starting, of course, with the patient and face value sort of stuff, symptoms and signs. In vitro and in vivo tests, if you know the difference between those. In vitro, if you draw blood from somebody and you do a test on it, it's outside of their body when you're doing the test. So that's in vitro. In vivo, if you guys got that tuberculosis PPD test, that was in vivo because it was on your body. So. Okay, cultivation and isolation. That just talked a little bit about selective and differential media again. and how that could be used, and the streak plate again, and how that could be used to isolate and characterize. We talked also in this one about Kirby Bauer, not just to figure out what antibiotic to treat your patient with, but also potentially some bacteria can be identified by their reaction profile to specific antibiotics. And then phage typing. Bacteriophages, each bacteriophage binds or infects only one species or even serotype of bacteria. So if it infects, your sample from your patient, and it only affects Salmonella, then your patient has Salmonella. That's phage typing. All right. All right, the end with that one. Thank God. And I say that, and I'm pretty sure that the lab one is not quite this.
I'll be a little competitive.
No, you're great.
It's the last part is my son.
All righty. So we'll do this. Let's see if I can get to it. Can't, it's gonna make it difficult, doesn't it? How do I get back to the phone? This is yeah. Oh, this is so hard to work. Classic mode. All right. Sign in for a minute. I don't know why I want to hear this one. I don't know if there's anybody new joining in on the Zoom side, but if you are, this is the lab one. Moving on to the lab one now. Let's just stay down now. Oh, sorry. All right, guys, I'm gonna go ahead and start it now. Remember, you can always join in as it's going as well. Just go to the Kahoot thing and enter in that pin. All right, so of course, I'm telling you guys that you need to review the safety stuff. It's basic stuff. Just be sure you go over and know the rules for the lab. There will be questions over that. Start with that. Where should broken glass be disposed? Honestly, I think you probably like this quiz better because their questions, I feel like this. Okay, so broken glass will go in that sharps container. Good job. Start to thing. right. So now we're going to get into the stains. With the stains, be sure you know your steps in order. You want to know each stain used and its purpose, why you would be doing it, what a positive would look like, what a negative would look like, and if something went wrong in those steps, what it would look like at that point. And what would it look like with a mixed sample? Because you identify a positive or a negative base, this sort of thing. Yeah, definitely know your stain. What is the purpose of heat fixing? All right, so the purpose is going to be adhering it to the slide and to kill it. Good job. You guys got that one pretty good. So what is the counter stain used in the Gram stain? The counter stain. All right, so the counter stain is safranin for the Gram stain, where our primary stain is going to be crystal violet. Mordant will be iodine. Then we're going to decolorize with alcohol and then counter stain with safranin. And remember that we'll get tested over that on both exams. What color will Gram negative be after the Gram stain? Don't let the pictures fool you guys. They're just pictures. Gram negative is going to be red. Awesome. What is the first stain used in the capsule stain? That's going to be Congo red. That's when we put the Congo red dot and mix in our sample and then spread it out and let it dry. Yeah, I didn't realize how many I put. All right, what type of stain is Congo red? All right, so that's going to be, it is a negative stain and it is a background stain as well. Negative, that just means that it has a sort of negative charge aspects to it. Remember, the outside of the cells do too. So it repels, they repel, and so it stains the background. So it's both. All right. So in the picture, this is a pure culture. So what stain is pictured? All right, so that's the endospore stain. The red are, well, I better not say because I think it's on the next question. We'll see. I think it is. Yeah, what is the term for the red structures in the image? So the answer is going to be vegetative cells. I had a feeling.
Only partially correct. I
Yeah.
didn't think the other part.
I don't. Which of the following bacteria would be acid-fast positive? So that's just going to be the Mycobacterium flea in this list. Most of you guys got that, so good job. What is the primary stain used in the acid-fast stain? Yep, so that's going to be carbofusion. In which staining procedure did we use steam? Yes, endospore stain. So that one actually used the steam. Remember that capsule used a little dunking poplin jar. Which of the following structures allow for bacterial motility? Select all that apply.
Only one needs to let one. Right.
Oh, did I put it? The one.
Only that one.
Well, you're welcome, I guess. I was like trying to be tricky. Yeah, it's only flagella. Whoops. So other ones are for attachment or like the sex villi for exchanging genetic information. All right, so the stains that you'll want to go over are going to be simple staining, gram stain, acid-fast stain, capsule, wet mount, which was not really technically a stain, but you wanted the purpose for it. And then the endospore stain. So know what dyes you would use or what stains you would use and in what order. You don't need to know the times. That's not important. But the stains in their order that they're in are usually concept based, So Eliza, when I say know this in and out, I'm more or less talking about like people get so confused about indirect and direct. So definitely get those under control. What does the E stand for? Just occurred to me, I wrote all that crap on the board and then we're taking the test in here tomorrow. I'll have to erase that. Right. Take a picture of it and I'll just redraw it, I guess. All right, so it stands for enzyme. Surprise. So it's enzyme-linked immunosorbent assay. So what is an indirect, Eliza, looking for? We had questions about this on the lecture one. I remember you guys doing pretty well at it. So indirect is looking for the presence of the antibody to whatever disease typically. So good job. Okay, so that's it for our ELISA question. Be aware of the principles of how the assay works. What are antibodies? What are antigens? I don't need too much detail about these. Just be familiar with the fact that antibodies are proteins made by the immune system that recognize specific antigens. And then antigens are anything your body can see as foreign, right? So indirect immunoassays versus direct immunoassays. So when we say indirect or direct, it doesn't only apply to ELISA. It's anything immune. So if we're looking for antibody or if we're looking for antigen in any kind of test, then we might refer to it as indirect or direct, respectively. So how the color change happens, that's just referring to the fact that you have the enzyme linked to that kind of secondary antibody that cleaves the substrate and causes a color change. If it's there, then that'll happen. If not, then you're negative.
I'm sorry.
Yeah, you're fine.
Can you, again, one more time define antibody and antigen?
Okay, you're fine. An antibody is a protein that your body makes as a defense against an antigen, a specific antigen. So antibodies and antigens, very specific binding going on. And then the antigen, of course, is anything that your body could react to as foreign with the immune system. That help?
Yes.
It's unfortunate because we're going to learn about antibodies when you guys get back. So I don't know, you missed out on that. Timing wasn't good. Our DNA stuff. The DNA stuff was all taken care of on the online junk. We didn't do it in class, but it will be on the midterm. Heads up. Right. What does PCR do? Lucky we kind of talked about this already. So PCR is going to it's going to amplify DNA. And the way it's going to do that is going from 1 and doubling that 1 every time. So if you made 1 and you got 2 as a result, then 2 would go to 4 as a result, and so on and so forth for every single round that you go through. So you can get even the tiniest amount of DNA and amplify it using. Which DNA fragment is largest? Everybody does terrible on this question. All right, so 35 kilobase pairs is going to be the largest here. So that KBP means kilobase pairs, which is 1,000 base pairs. If it's just BP, that's just single base pairs. Okay, so if you see K, then add, it's 1,000. So yeah, that's going to be the biggest one. Did better than last semester. All right, which of the following DNA fragments would travel the farthest on an agarose gel? Okay, so the answer for this one is going to be 100 base pairs. Most of you guys got that. So the smallest ones are going to travel faster. Think of it as little kids squeezing through to get out after a Thunder game, as opposed to like a sumo wrestler. All right, so this is more stuff about the DNA that you might want to be aware of. The DNA structure and its function. The fragment sizes, that's talking about, comparing those kilobase pairs, the base pairs, and how they would run on the gel. The agarose gel electrophoresis principles themselves. Steps and components for PCR, bacterial transformation, which you guys did do that online thing for. We normally have an experiment associated with that, but I usually cut it out if I'm being completely honest. And then plasmids and what they are and what they can be used for. A lot of the stuff you're covering in your lecture stuff too. So
can you elaborate on the one that was bacterial transformation?
Right.
I did them, but I don't.
Yeah.
Can't think of which one it is.
So bacterial transformation is whenever you have a competent bacterial cell that can pick up DNA from its environment. So a lot of times what we would have done in our experiment, we would have had a plasmid, and encoded in that plasmid, we would have antibiotic resistance, and we would have had glowing green stuff. Okay, glowing green stuff would be associated with the presence of something called arabinose, which is a sugar, the presence of sugar, right? That special sugar. And it would only make that green if that sugar was there. So cool. So that's what the plasmid coded for. We shoved it into those competent cells, usually by hot and cold, it shocks it into their system. Then you plate all of the bacteria that you have onto the antibiotic plate. If you have colonies growing, they have to have the plasmid in them because they wouldn't be able to grow without the plasmid. The plasmid gives them antibiotic resistance. Does that make sense?
I think it's the, yes, and I'm trying to think it when we're doing it, it's the one where you had to put them in the electric, like if we were. I think I got it.
I don't know how it looks on the online one specifically, but yeah, I mean, you would just basically be taking, like again, in our lab, it would have just been hot and cold shock. But yes, there's other ways that you can do it that force it to happen. You can use electricity, but getting competent cells to take up DNA, that's the big message. I'm not going to ask about any specifics outside of that. But I would be aware of that plasmid, you know, what we talked already about what plasmids are, but be aware of it. The way that that worked, we use when we plate that on the antibiotic plate, only the cells that have the plasmid will grow, but they won't be green until you give them a special sugar. Then they'll be. That's important too. So, all right, that's plasmids and transformation. Next thing. Eukaryotes. Review your drawings, and I can't stress it enough. Make sure you focus on the differences. All right, here's some quizzes. Which organism is this? It's hard to see on that screen. It's easier on that back screen.
To tell you this all, yeah.
It's okay. This one is one you just have to kind of memorize. All right, so this was penicillium. Again, memorize. Like, I don't know how to describe that to you otherwise. All right, what is the name of this organism? Perfect. You guys did recognize Giardia. I couldn't have shown it more, honestly. I'm glad you at least saw that one. All right. Look over the organisms you had to draw. I'll be able to identify them based on their differences, basically.
Is there going to be a lot of questions about that?
There'll be a few. It's not going to be like attribute test or something like that. I don't like those questions. I want to say there's like maybe 2. 3 tops. Like, I don't think there is 3.
Like, there are main ones that we should look at.
Well, like I said, it's not going to have any cysts. That's already cut it down some for you guys. Other than that, I think I say the protozoa, but that's like most of the thing. So, I would just focus mostly on the protozoa. I know that doesn't narrow it down a ton, but protozoa, don't worry about the cysts. So I guess the trophozoa is mostly, but yeah. Well, that's not all that's going to be on there. I didn't want to be clear, but I mean, to me, I feel like the others, I don't know what else to say about it, but yeah, there's not going to be any cysts on there for sure. And well, there are eggs. I mean, we look at a lot of the eggs from the helminths. Just be aware of the differences there too. Trying to think of anything else. I can't, but I will say this crap's going to be on the midterm, but it will not be on your final. So just slide through this one and it will be okay. All righty. general bacterial growth stuff. So media concepts, bacterial growth, culture techniques, that junk. Which is not a term used to describe colony morphology. All right, so the answer is hyphae. Hyphae are a structure in fungi, but it's not colony morphology. All right, what is the name of this type of culture? It's hard to tell in this picture, I apologize. Look at it closely. Yeah, I'm glad you could tell. I felt like it was a terrible picture. Yes, that is slant. How was this organism isolated? Yep. Cool. All right. We're getting there. Your favorite. Which arrangement or arrangements of bacteria do you observe? Now, this is a little tricky, so just pay attention to what's going on here. Try not to leave something in. I was really inconsistent with capitalizing. All right, so the answer is going to be Diplo and Strepto. Diplo, there's like those two that are like off to the side, but technically that is Diplo. And then Strepto, it is in chains, and you can see how the chains are actually folded kind of back on each other, but that was just Strepto. Cocci doesn't count because that's not an arrangement. That's a cell shape. Yeah, that's the problem. Okay. So this one might be a similar thing. What is the name of the shape of these bacteria? I'm sure you won't get caught again. This one's a little harder. Oh no, it's not bad. So yeah, this is spirillum, which is just kind of spiral shaped, roughly. You can kind of tell just by the S shape of it. All right, that was fun. Bacterial growth. You want to look over your colony morphology terms, your media types, and that's just talking about like broth, plant, plate auger, that sort of stuff. Isolation techniques. We mostly did the street plate, but you can do dilution as well.
The study guide, it says which one to use for.
Definitely the street plate.
Okay, yeah, I know which one we use, but I.
Yeah. Yes. So the one that's used the most is definitely the street plate. That's the one you want to lean towards if you have choices between them. Cell shapes like bacillus and cocci, and the spiral is basically pretty much the only cell shapes you're going to see. And then the cell arrangements, Strepto, Diplo, Staphylo, the tetrad, Sarcina, Palisades. I think that's it. Oh, geez. Okay. Microscopes. This should be easy. So, if you observe a specimen at 40x objective and have a 10x ocular lens, what is the total magnification? So the answer is going to be 400. Just multiply them. So which objective lens do we use for oil immersion? It should be easy for you guys. Perfect. All right, that's all we're doing with the microscopes, I guess. So calculating the magnification, understanding how they work, the parts, how to use the parts properly, and oil immersion and what it's for. Next is Kirby-Bauer. So we're looking at this one. It's going to be antibiotic susceptibility and resistance. Which antibiotic is this bacterium least susceptible to? So the answer is definitely going to be E. So in the past, we did this through PowerPoint, like a PowerPoint quiz with the clickers that you guys don't have to use. But this question was a all or nothing. You could like bet like if you wanted to put in all your points or not. And so, you know, people would often like put in all their points and then lose them all. They got the wrong answer. I can't do that on this. It would be fun though. All right, Kirby-Bauer, know what the zone of inhibition is. The MIC is a minimum inhibitory concentration. So the minimum amount it would take to inhibit growth. Mueller, Hinton, Auger, we needed it to be uniform and the pH controlled. So that can control the spread of the antibiotic in the media. So it's even, as well as pH not affecting it. We know how squirrely those hydrogen ions can be. And then bactericidal versus bacteriostatic, which again, are terms that you need to be familiar with for the lecture as well. So I think this is it, right? Yeah. So if you guys have any questions, let me know. Have fun studying. I hope that helps you guys figure out maybe some topics that you need to go back and cover again. So if you had a chapter you did particularly poorly on, definitely focus more time on that than like chapter. I'm imagining everybody did pretty well in chapter 1. Here's our results, right? I forget about this part.
The last question, I solved it absolutely wrong. It's the opposite.
That was really close, so. Righty, all righty.
You be on this test, and I did. Winning.
I will post these online. I'll try to edit those questions on the other one that like I don't know what what I'm thinking, but I don't know what all of them were. We'll try to find them and update them.
Was that a good portion of what's gonna be on the lab?
No.
Okay.
Yeah. And I'll say that like I don't know. I really do think people think I'm exaggerating when I say people do bad on this test. I'm not exaggerating. Like this is a 48% was last. But just in case the kind of plan that we have for this exam, come in, sign in, you get your paper test, you sit down, you basically don't move from that spot unless there's some sort of actual emergency. I mean actual. And then you stay there and you take your test. And then when you're done, you'll hand in your test and I will see that you have a grade online and you'll be good to go.
You do them at the same time?
Yes, well, you can. I mean,
Like one is paper and the other one is online. Is that at the same time, or do I come in and take my online and then?
so I'm going to give you, you can do both. Like you come in and essentially like you'll have the password to do the online and you'll have the actual lab test. And I don't care which one you do first or second, because like there's a lot of overlap and I understand that. So whichever one you want to do first and get out of the way first, I'll leave that up to everybody in their own decision. And then so you'll do one and you can wait and hand in the lab one if you want to, if you think that's going to help you somehow and look back at it. But I don't care, because to me, it's just, you know, the same material, whether it's on that test or the other test, so who cares? But yeah, so that's how it's going to be. I feel like it's pretty generous, but if you have any confusion about it, of course, beforehand, just email me. I'll be here at 9 too, so if you show up and you have questions about stuff, just let me know. Bring your laptops. I posted the link for Respondus. Please be sure you download it. Differences in augers, like the are you asking about like the selective media? Yeah, no, you don't have to know that. We haven't actually learned that yet. So that's what we're going to be doing when you guys get back. So general media, just like, you know, slants and broth and stuff. The differential and selective media, we'll do that when you guys get back. So we'll start with the, you know, hardcore with the unknown stuff. But yeah. Yeah. All right. All right. I don't know. I don't try to make it dismal. I know I do it every time. Sorry. Maybe I'll put like jellyfish on too. And that's not exciting, so I'm just talking to myself. But what's up?
I just have a question about.
So differential diagnoses are, that's basically referring to other diseases that you might think it was in your diagnosis. So like if you, yeah, it's like if you see a patient comes in with a flu, your differential diagnosis might be COVID. There could be several. That makes sense? Yeah.
So my physiology test was yesterday, so whatever. It was,
Uh-huh.
yeah. It was right after lab. So I was gonna let you know that I was here in here on the testing center.
Yeah.
So that's fine.
I'll take care of that. Don't worry about it. Yeah.
Cool.
It's gonna be a lot easier on you to be in here.
Oh, yeah. Thank you.
Yeah. Yes, yeah. Right, guys.
My
you wanna-
Is
what's up?
where is the testing center at on campus?
Testing center is in, it's like if you go to the hub, but go downstairs, you'll run right into it at the bottom of that round staircase.
Okay, perfect. Thank you.
Yeah, it's on that lower floor. You guys have any other questions online, folks? All right, I'm gonna cut you loose then. Thanks for joining, guys. So